Hi everyone, and welcome to this demo for Pf-HaploAtlas. The Pf-HaploAtlas allows anyone with an internet connection to study genetic mutations in any of the genes of the Plasmodium falciparum core genome. The specific combination of mutations in a gene is referred to as a haplotype, and this is where we get the name HaploAtlas. Pf-HaploAtlas uses data from the malaria gen Pf7 dataset, and this means over 16,000 parasite genomes are being used to help you study evolutionary trends for Plasmodium falciparum. Let's start by choosing a gene. You can do this by coming over here and scrolling through the list of genes in the genome and choosing any one that interests you. In this demo, we'll be looking at Couch13. You may know that mutations in this gene have been associated with resistance to the anti-malarial drug artemisinin, so it will be an interesting gene to look at together. Once a gene is selected, the app generates the first of 3 plots. This plot is known as the haplotype upset plot, and it gives you a broad overview of the haplotypes that were observed for the gene selected. If we take a closer look, you'll see that there are 3 subplots stacked on top of each other. In fact, each of the columns of data in this plot represents one haplotype, and each haplotype comes with 3 pieces of valuable information. Firstly, we can see how many samples in the data had this haplotype. We then can see the geographic distribution of the samples containing this haplotype. And finally, we can see the mutational composition of this haplotype. So let's put this into practice for Couch13. By looking at the first haplotype here, we can see that there have been 6,899 samples observed with this haplotype. And going down, we can see that there is a broad geographic distribution, as demonstrated by all of these different colours. The mutational composition section for this haplotype shows that there are actually no mutations, since there are no black dots here in this column. And that's because this haplotype is the same as the 3D7 reference lab strains haplotype, and so there shouldn't be any mutations here. Now, let's take a closer look at the third haplotype here. This haplotype was observed in 1,811 samples. It was predominantly observed in just one geographic location, as you can tell by the dominance of this dark blue colour in the bar. If we scroll over to the sidebar here and move down, we can get some more information as to what this location was. So this dark blue colour was the eastern part of Southeast Asia. Finally, the mutational composition shows us that this haplotype consists of just one amino acid difference from the 3D7 reference. This haplotype has a C580Y mutation, which means that in these parasites, the 580th amino acid of the Couch13 protein is a Y amino acid compared to the usual C, found in 3D7. This means a cysteine is replaced with a tyrosine. This C580Y mutation in Couch13 is actually very important, both biologically and clinically. And it's been on the malaria community's radar for some time now, as it's one of the mutations that provides the malaria parasite some resistance to the drug artemisinin, which is part of the first-line treatment recommended for malaria by the WHO. This haplotype will be interesting to investigate further, and we can do that by clicking on any of these 3 data elements belonging to this haplotype, like so. And it will generate a further 2 plots specifically for the haplotype you've selected. So after clicking on the C580Y haplotype, we were prompted to scroll down to view the new plots which have been generated. The first of these plots is called the Abacus plot, and the Abacus plot shows how the haplotype frequency of your selected haplotype has changed with time and space. A key component of the Abacus plot is what we referred to as beads, and these are these circles you can see on the plot. Each bead tells you that there were samples collected in this location in this year. The absence of a bead means no samples were collected in that location or year. The beads are coloured, and the darker the colour of the bead, the higher the proportion of the samples here containing the haplotype you've chosen. What we're ultimately looking for in the Abacus plot are gradients of colour as we go forwards in time, so towards the right. And you can see there's something clearly going on here in the eastern part of Southeast Asia, as generally the plot is growing darker as we move towards the right. So the prevalence of C580Y is increasing with time here. For example, in this part of Thailand, there were plenty of samples collected which didn't have the haplotype, but we see it start to emerge from 2011 onwards, and you can get details from each bead when you hover over them. Similar has happened in Vietnam. C580Y was found at very low frequencies before shooting up to around 80 or 90%. Finally, in Cambodia, we see a gradual increasing gradient of colour from around 50% in 2010 to 80 or 90% in 2017. Ever since the introduction of artemisinin as an anti-malarial drug, parasites have been under increasing evolutionary pressure to survive the effects of the drug, and this C580Y mutation is a well-studied mutation which helps the parasite to do just that. So here you can clearly see how Pf-HaploAtlas is useful for tracking known drug resistance mutations. Now I'll introduce you to the last of the three plots, the world map plot. This plot displays the average frequency of the haplotype over an interval of time of your choice, and this is done at a country level. Similarly, as before in the Abacus plot, the colour intensity of each bead corresponds to the haplotype frequency. On the world map plot, the beads have got a ring of colour around them which indicates their geographic distribution. And remember, you can check the sidebar to see which colour corresponds to which geographic population. The world map plot helps you to get a better idea of where the haplotype trends are in relation to the whole globe. Great. So now we've walked through all of the plots generated by Pf-HaploAtlas for a given gene, and I'll go on to showing you some additional features of the app. So by going back up to the top and clicking this drop-down button, we can firstly see that Pf-HaploAtlas allows you to decrease or increase the minimum number of samples per haplotype used for analysis. You can also view some statistics as to why certain samples were excluded from the analysis. A great feature of Pf-HaploAtlas is that you can download the underlying data in analysis-ready aggregations. Here, you can choose from two different types of aggregation if you wish to use the data for your own analysis. Finally, you can change the rendering of this first sample count plot by choosing to view the y-axis on a log scale if you wish. If the gene that you're studying is particularly interesting to you and you want to know more, you can continue to browse it on Plasmodium by clicking this button here. You can also download any of the three plots by clicking on one of these orange buttons to download it in various file formats. Finally, if there are any features that you'd like to request for the app, you can go to the sidebar and contact us using any of these options here. Brilliant. So we've now covered all the basics for Pf-HaploAtlas. Let's move on to another interesting example. For this second example, we're going to jump to another gene entirely. This gene is called AAT1, and we can search for it by typing it into this search bar. The haplotype upset plot is quickly rendered for AAT1. So AAT1 has only very recently been associated with resistance to anti-malarial drugs, so it's not quite as well studied as the Kelch13 gene we just looked at. AAT1 is a good example of how Pf-HaploAtlas can showcase important and interesting evolutionary trends in lesser studied genes. If we look at the second haplotype of AAT1, we can see it was found in 1,855 samples. It was predominantly found in the eastern part of Southeast Asia, and it was made up of two mutations, F313S and Q454E. As before, we can dive deeper by clicking on this haplotype and bring up the Abacus plot. On the Abacus plot, we can see a distinct colour gradient in the eastern part of Southeast Asia. And this indicates some evolutionary pressure on this haplotype, which is of interest to us. You can read more about the biological significance of this example haplotype in the Pf-HaploAtlas scientific manuscript. You yourself might be able to imagine browsing Pf-HaploAtlas and stumbling across similarly understudied genes with possible signs of evolutionary pressure, such as this, to help you in your research. So that's everything. Thank you very much for joining me in this demo for the Pf-HaploAtlas.